Hello, everyone. I see our participant number rising, so I'm going to let it get continue to increase, and we'll get started in just a moment. It is great to see everyone joining us on this Wednesday afternoon or evening, wherever you're tuning in from. For the East Coasters, it's certainly dinner time, so we appreciate your presence. All right. I'll give it another minute. I'm so excited to have this session tonight that I don't want to rush us, but I also know we have a lot to get through in the next hour. So I want to use our time wisely. And it looks like we're a couple minutes past the hour. And so, um, if if I'm missing anything, we'll have to circle back. But I think it'll be maybe just a good time for me to introduce myself. Um, My name is Dr. Natalie Exum. I am an assistant professor at the Johns Hopkins Bloomberg School of Public Health. My training and background is both in environmental engineering and hydrology and how water moves through the environment and contaminants move through that water. But I also have a PhD in public health from Johns Hopkins, where my work is always focused on water systems and linking water systems to public health. And I have a laboratory where I focus more on environmental microbiology, but work largely with communities and their concerns about water contamination, mostly in the Potomac watershed. And that brought me to work closely with the Potomac River Keepers, and I'm now a member of their PFAS expert advisory board. And so that's brought me here today to monitor this session for you, and just delighted to get us kind of kicked off. And um, I will introduce our speakers kind of as they're coming up. But just to reiterate for us today, we are here to talk about the health effects of these substances. And let's just jump right in. So I'm just going to set the stage for us for this one specific class of contaminants, and really just talk about their health effects. And so as we say PFAS, what we are really meaning are per and polyfluoroalkyl substances. And this carbon fluorine bond is one of the strongest that there is out there. And so it makes them very hard to break down in the environment. It makes them very good and very stable for putting into firefighting foam and Teflon and all these substances that we know that they are now in. And so for the chemistry nerds out there, that's what this compound looks like. So, I think it's important for us to get started by saying that, the science has always been behind these chemicals getting into the environment. Right now, you're going to talk to a slew of scientists here. We really want to focus on the facts. But, the reason why we're in this predicament we're in right now is because 40, 50, 60 years ago, 3M and a number of other different, DuPont made these chemicals and said, we are going to make them where they are not going to get out into the environment. And that was kind of how they sold this as these data, when they very well knew that these were going to have, that these were dangerous for health. Right. But now here I'm going to talk to you. That is a story that is well told, and they're out in their environment, and now they are circulating in many different ways. And so, I'm not going to go through all of them today, but the one we're really going to focus on today, because there has been a lot of progress made in terms of getting regulations through for the Safe Drinking Water Act to regulate a certain, six certain PFAS chemicals. But this biosolid space, and how we have used sewage sludge to spread across our fields and our farms across America, really has not gotten as much attention. It's also not regulated. And so that really is what we're here to talk about today. And, we're just going to breeze through these things. And so really, how are people exposed? We really think about this through drinking water, food, and then, dermal contact, right? So if you're, if you're wearing some sort of waterproof clothes or such, or even breast milk or cord blood, right? So these are in when we look at human blood levels, this is in 97% of all Americans. So we all have these in us, and as we say in environmental health, it's the the dose that causes the poison. And so we really are going to talk about the concern with these places where we have really high levels of them. So just to say, there's PFAS water contamination in many sites. This number comes from 2021. The number has certainly grown since then, but it truly is everywhere. Where you have regions where there's military sites or airports where there's been more of these firefighting foams that have been used, you tend to see higher levels of them in groundwater as well. But these drinking water, they're detected in many drinking water systems. And these are just the ones that we were looking for a certain class, right? We're not going to talk about long chain and short chain, or maybe we will today from your more of your scientists that are coming up. But just to say that this is certainly an underestimate of what you're looking at on your screen right now. Now, as we go through the science, I really want to talk about a really key report that came out a couple years ago from the National Academies of Sciences and Engineering Medicine. And what the National Academies did, which is very useful exercise for us here today, is it's a consensus report. And they got experts in the room to talk about the existing studies for what we know about how these chemicals are impacting health, right? You can go through the list, but we really want to stick to the science and how good the studies are to show what's happening. And so as you'll see here, they went through a very in-depth process. You can look it up if you're more interested in how they got to these findings. But they did note that the health effects span many different categories, okay? And they're associated with many different health effects, and this is now true from a number of different authoritative bodies. So we know these are important for health. The ones that we have clearly sufficient evidence for, and the ones that we have suggestive evidence for, are listed here on your page. Um, you know, uh, Certainly they cause a decreased response to, say, vaccines, right? So you have your antibody responses are decreased after, if you've been exposed to these, so clearly there's kidney cancers that are linked to these, decreased infant and fetal growth, and this kind of dyslipidemia, which is, a lipid dysfunction. So we know that that is certainly clear. These other ones that are suggestive, meaning like the science is just catching up, right? And when we did this consensus report, this is what we saw: thyroid disease, ulcerative colitis, breast cancer, testicular cancers, pregnancy-induced hypertension rates of preeclampsia is a huge risk for pregnant mothers. The list for, I want to say the list goes on, but as I'm sticking to the science, this is what this consensus report gave us. And so how to think about, reducing exposure, right? You can have an in-home filtration system that you put on your faucet, right, to reduce them if you think that you have them in your drinking water. There's ways to change your diet to not, but really it's very challenging if it's very challenging to know where are these coming from if you end up getting a blood lead test. And that brings me to one of the most important findings from this report, and I'm going to pass it off um, after this. So this is now a recommendation for clinical care. Okay, the state of Maine is actually taking this up and recommending that if you are concerned, right, if you're a farmer and you know that this has contaminated your field and you're concerned for your family, you could get a blood test in Maine, and if you come up within one of these ranges. There are recommendations in this report for what to do and how to think about reducing your exposures. And so this is a real revelation. We do this all the time for, blood lead, right, for our kids. It's very clear. We have clear brackets, but this is our first attempt to, at a population level, say, okay, the levels in this community are too high, and these ranges were established in this consensus report. So just to offer that here, to start us off, to say we know that this is important for health, and we know that for the future of our food supply and our water supply, that we need to be addressing how these chemicals are getting concentrated and becoming a source at our wastewater treatment plants, and then how those biosolids are put out into our fields and our farms. So that is our start for you. I am now going to introduce Dr. Omogamre, who is an assistant professor of biology and environmental toxicology at the University of Maryland Eastern Shore, where he leads the environmental toxicology and intervention laboratory, and his research investigates the ecological impacts of PFAS in agriculture and natural systems using phenotypic, biochemical, and molecular approaches to probe plant and insect models. Very interesting for us today. And among his lab's priorities, he's uncovering the molecular mechanisms and environmental conditions that drive the transportation, right, the movement of these PFAS and the accumulation in plants with the aim to optimize the phyto extraction for remediation, right? We need to be thinking about remediation in all of this. In non-food plants, and also advancing, and reducing the uptake in food crops while maintaining crop resilience under exposure. And he's also advancing functionalized biochars that improve PFAS immobilization and limit bioavailability. And his real goal is to bridge the mechanistic discovery with the practical and future-ready solutions to help farmers, communities, and policymakers confront PFAS challenges. So we're delighted to have you, Dr. Montgomery, and I know you may need to leave early, but off to you.
Thank you. Appreciate that. And it's my pleasure to be here. When conversations about PFAS, I think that conversations I love to have because this is a challenge that is unique, and we all it would take a village literally to figure out the solutions. So really today I'm just going to talk about our understanding from our lab perspective of the PFAS challenge. Looking at the ecosystem level ecological problems, which eventually also connects to us as humans. We're part of the ecosystem. Again, like Natalie said, I am the PI of the Envitoxin lab at UMES. Our university is a land-grant university, so research around PFAS and farms and supporting farmers is something that we're really interested in. So I usually like to start my conversation in just, we all know what PFAS are. The numbers of this group of chemicals varies depending on the agency, the federal agency, or who you ask, right? But technically, we're talking about some 10 to 15,000 group of chemicals. And what differentiates them is the number of carbon chain length they have, the head group. But the general chemistry is similar. You have this hydrophobic tail, and of course, you have the hydrophilic head group, make them to act like surfactants. Of course, because of the properties, we see they're using several, the ubiquitous, using several applications, very stable to temperature, to chemicals, just name it. And so that's where the problem started. Because of the utility, their properties is now resulting in the problems that we're seeing today. Generally, with chemicals, synthetic chemicals, microbes in the environment typically help us to break them down. But the unique challenge with PFAS is we don't have that biodegradability of them. And so that's why we know them as forever chemicals. But the question that, why are microbes not able to break them down? Why are we in this situation? Mostly we hear about the carbon fluorine bonds, which is one of the problems. Of course, bond energy is so strong. And so microbes, the enzymes involved, it requires a lot of work, energy to kind of break that. But that's not the only problem. So if the microbes even succeed in defluorinating, cleaving out the fluorine from the structure, the fluoride ions are toxic. All right. So usually that's why you see studies today where they would report that it appears that there's some level of biotransformation, level of biodegradation, and then it stops. The understanding is as the fluoride level builds up, it begins to affect the enzymes and the microbes that are involved in the process. So this is one of the major challenge. And then if we go to the route of mineralization to kind of hit it, we're talking about some 1,000 to 1,200 degrees Celsius. So, of course, today the technologies that are in the works, supercritical water oxidation, that can mineralize PFAS. They've been tested in a lot of Department of Defense sites, but it's for water, all right? But a core problem is the land soil. We don't have solution for that currently. So And of course, the PFAS problem is unique because PFAS just it cycles in the environment. Of course, the whole biosolid conversation is there because, of course, wastewater treatment plants concentrate them. We've been using them for many years. We didn't know because our thinking is microbes would chew up every chemical. And so that's the understanding until recently. We know it's been accumulating in farmlands that have used biosolids for many years. And of course, it cycles back to the water through runoff. Plants pick it up, and of course, we eat it. So the cycle continues. So this is a challenge. So because of this challenge, personally in my lab, we're really interested in three things. First, how widespread is a contamination, especially in the lower eastern shore. That's really where we do some of our contamination work. Then also, what happens when organisms in our environment, from plants to beneficial insects, right? When they pick the exposed, what happens? And what can we do about the problem? So really, that's the three-pronged approach that my lab kind of uses in our research. So I'll briefly just tell you about some of the things we're doing. In no way does it cover everything. We have just 10 minutes. I just wanted to let you know some of the things. So let me start by telling you about one of the ambitious projects we're working on. Of course, recently, this project was driven by I live in Salisbury, right? So I don't know if you follow the news. There was a drinking water contamination, and there was a connection to Purdue plant that has impacted several homes, hundreds of homes. And so, so my question is, so right now, the understanding is that region has some elevated level of PFOA, PFOS in drinking water. So the question is, how widespread is this? Because the focus is just on that region. The eastern shore, lower eastern shore, you have a whole lot of biosolid applications. So it's good if we can do a systematic study. So right now we're trying to do that. We're collecting drinking water. We're using a community science approach where we're working with high school students. So we design the kits. We administer a curriculum, and then students collect it. We take it back. We do the analysis in our lab. Our goal is to use this approach to kind of map PFAS data using zip codes. We're also collecting metadata. Where are the homes getting the water from? Is it a well? Is it a city water? We're also checking how old is the house, right? We're collecting a lot of metadata just to see if there's contamination, what is driving that contamination. It's early, but we've had some data come in, and it's the early insight we're seeing is 75% of the water samples we've analyzed did show some PFAS. All right, the range is from 2.8 to somewhere around 14 parts per trillion. Now, interestingly, PFOA was showing up in 75% of the samples we've analyzed. PFOS is in 50%. However, their levels are below 4 parts per trillion. What we noticed, the 25% that didn't have PFAS, those homes used water filter. And so that goes back to what Natalie said earlier about, faucet, using those water filtration systems. It does help. It's early. We're still collecting data, but our goal is to map this so we can get to over 500 to 1,000 homes. We're using funds on our campus. We're hoping we're going to get funds to support this. But we believe that this study is really going to throw up some insight into drivers for PFAS and how we can reduce exposure. So those are some of the students working with me on this project. So on the environmental contamination level, I just wanted to share just this one study we're currently running. Now, the exposure impact is one of the big things that I am interested in. All right. So it's in the environment. What is it causing? All right. So currently I'm working with plant systems. Soybean is a model system I use for many of the experiments we're running. Our goal is to expand to other plants, which I'll talk about shortly. Then the armyworm species, just because they are critical pests for soybean and, of course, other crops the farmers grow in the Eastern Shore and, of course, in other parts of the United States. We're also interested in mosquitoes, and I'll tell you why we also started getting interested in mosquitoes once I show the next slide. So our goal is to understand what would PFAS in the environment do to plants, and of course, eventually, how does that affect insects that live off such plants, and of course, try to understand if their molecular signatures that support whatever phenotype we see in these model systems that we're studying. So let's start. I'll start by telling you about this study. So this is one of the signature studies we've done. We looked at a farm. It was hard, and I hope that at the end of this conversation, we could have more farmers who would be willing to let us do some more studies. So this farm has a history of biosolid use. So one thing we looked at, and for this study, we worked with the Choptank River Keeper, very, very supportive for us to get the data. So we analyzed PFAS in the soil, in the farm. We also looked at runoff ditches, and then we looked at the irrigation ponds where water runs to from the farm. And what we found is quite interesting. And I say that because it answers a critical question of how do we know the impacts the PFAS we're seeing is coming from the biosolid and not from runoff or rainfall or some other events. So generally, what we saw in the soil is we saw about 14 different PFAS. And most times, once you start seeing PFOS and you're seeing precursors for PFOS, usually that's a signature that comes from, biosolids. And most times, of course, the long chain PFAS, you see those also coming from biosolids. Short chain PFAS are typical, of course, the ones you find in the air that get dissolved from rainfall, are mostly short chain PFAS. Even if you have adsorption to particles, you don't see that effect very much. So the signature we see from here compared to what literature suggests is this impact is likely from the biosolid. And then we tracked in the water, we also we saw some close to 2,600 parts per trillion total PFAS, right, in the soil. Now, when we got to the irrigation ditch, that kind of dropped some tenfold to somewhere around 300 parts per trillion total PFAS. And the number of PFAS reduced from 14 to 10. And eventually, when we check the irrigation water, you also see a drop. So what we did see is that the trend, the pattern just suggests that the biosolid impacted farm runoff is transporting the PFAS, because most times we're seeing more of the carboxylates, more of the short chain PFAS showing up eventually in the irrigation pond, which makes sense. The long chain PFAS most times typically bind a little more to soil and they don't travel as easily as the short chain PFAS. So this kind of helped us to understand the cycling of PFAS from biosolid use in the farm. So the next question we were now asking is, what if the farmer decides to use the irrigation water in the pond for growing their crops? What could be the impact in terms of accumulation? So we kind of simulated that in the lab. So in the lab, we designed a simple study where we had the similar mixture that we saw in the irrigation pond. We spiked water sample at different levels. One is relevant. It was around total PFAS was 250 parts per trillion, which is under close to what we're finding in irrigation ditches. And then we had a very high concentration just to see if the dose does play a role in whatever effect we see in accumulation. Well, for this, I'm just going to show you some of the accumulation data we saw. Now, for the 250 parts per trillion group, we did see that in the root region, the 6-carbon carboxylate, HPA, 7-carbon carboxylate, and we also have the sulfonate, right, showing up in the roots area for the low treatment group. But for the high treatment group, you have a slew, almost all of the PFAS in the water eventually ended up in the root system. But when we go to the beans, which is more critical, we did see that more of the short chain carboxylates, all right? We had PFPA, PFPEA, we had PFBS there too, and of course, PFHX8. They showed up in the beans, all right? For the low treatment group, which is environmentally relevant. So this is, of course, this data, the data we found has been supported by other studies that show that when you grow, of course, crops with contaminated irrigation water, there's a possibility that the area parts are going to get contaminated. Or, of course, in this case, the soybean, we're seeing that the PFAS is showing up. And the signature in the low treatment group and the high treatment group are kind of similar. And it makes sense because, of course, the shorter the chain, the more hydrophilic, and of course, they can travel to the area parts of the plants much more than long chain PFAS. So this, what we found here, along with what we're seeing in the field, kind of now leads us to much of the next study I'm going to quickly talk about. We're seeing that short chain PFPA is the most highly concentrated in the area parts. it now led us to ask the question, if PFB is accumulating in the leaves, and of course, insect larvae, like fall armyworm larvae chewing on the leaves, what happens, right? That's why we designed this study. And what we did see, we now kind of simulated in the lab with spiked artificial dyes with PFBA across a wide range of concentration. What we saw is in the environmentally relevant region, we're seeing there's some level of stimulation. And we also confirmed that when we took leaves from plants that had PFBA, we fed them on this larvae. We did see that it has stimulated development. And eventually, of course, we had a rate of growth here. You may not see it. We saw, in the low treatment group, we were seeing like 70, they were getting 70 milligram per day. The controls are somewhere around 40 something. And of course, the high treatment group were somewhere around 20 milligram per day. So of course, because of time, what we're seeing here is there appears to be stimulation. We checked how much diet they were eating. We did see some 20% increased diet. We looked at that also, which kind of compares to what we saw with the leaf area damage study that we did. And when we followed that up with molecular signatures, we saw genes that did show that the stimulated growth is supported by genes related to cytoskeletal sheddings and all other metabolic functions. So we're running. Yeah. So got 2 minutes.
Yeah.
All right. We also did something with mosquitoes because, of course, we were like, okay, if there's PFAS in the irrigation pond, mosquitoes developing, what would be? We did also see some kind of stimulation in their growth based off our lab studies. I mean, I don't have time to go into all that, but what you're seeing here is the stimulation. The high treatment group, you don't see that, but those close to the controls, we see that. So these are just some of the exposure experiments we're doing. We're also doing remediation. So the question is, are there things we can do to immobilize PFAS in the soil? So we have some experiment going on. We have some promising materials with functionalized biochar. It's been effective where we have filed a non-provisional patent to kind of begin to develop that to eventually get it to the market to support farmers. So just to kind of summarize final words, our goal, like I would say, some people think, well, we're trying to get the remember, we're trying to get the farmers, lab. No, for me, our lab, we're trying to support. This is a problem we all want to solve. So the more farmers are able to collaborate with us in research, the more the chance that we can find some solution to this wicked problem. So really, that's my 10-minute presentation. I apologize for going a little over time.
Never apologize when we're talking about solutions to really understand where, I'm excited for the research you're doing, especially on the Eastern Shore. And I did hear about that Purdue case, so glad you're out there. Thank you, Dr. Montgomery. So next up is Dr. Stephen Lissay, an environmental toxicologist that runs an consulting firm called Lissay Research and Consulting, and he primarily works on cases involving PFAS contaminating and advises laboratories on developing PFAS extraction techniques, not easy stuff, and obtaining PFAS analysis certifications. So Dr. Lissay developed modeling equations that are used to predict plant accumulation of PFAS from the soil. Really important stuff. If we know we have PFAS-laden soil, what should we be growing on on that land? So glad you're doing that. And these equations have been used to develop state bills and county ordinances. All right, your turn, Dr. Lissay.
Thank you very much, Natalie, for the introduction. All right. Can everybody see? Can we see my presentation just fine? Thank you. All right. So we're talking about biosolids. What are biosolids? Well, they're the solid leftovers from a variety of different treatment processes, waste treatment processes. And the EPA categorized them in two major ways. There's Class A biosolids and Class B. Class A, there's strict pollutant ceilings and cumulative limits that are in them. As Natalie had mentioned just earlier, that does not include PFAS, so there are no limits on PFAS in the products. And Class A can be sold in bags in retail stores. Typically, they're used by everyday people for their lawn, garden, public parks, whatever. Class B, on the other hand, are a little less treated. They have more lax limits than Class A, although they have the same limits. And typically they have more restrictions on their use, right? They have crop harvesting restrictions, right? So you can only apply them and only after so much later you can actually harvest the crop. And also after you apply them, there's site access limitations, so you can't go to the site. Additionally, they can't be sold in bags to the general public, and most of them tend to be bulk applied to agricultural lands, forests, and reclamation sites. And so here's an image of Class A biosolids. I took this from the company's website. Typically, they are widely available, as I had mentioned before, and they're not often visibly labeled. You typically have to go to the back and find, derived from biosolids on the back of the product. And this is by no means one of the products. There are several of the products, and several of these products can also be combinations of biosolid material and non-biosolid material. And then Class B biosolids, it's more typical to see large tractor trailers on lands applying this everywhere. This is more of an industrial scale type product. And typically you can smell it from miles away. All right, so we had mentioned and stated that biosolids tend to have PFAS in them. Well, how much PFAS in them? It's kind of really dependent on the product, right? It depends on where that waste that was used to make these products is derived from, right? So this is a study out of Linda Lee's lab showing they tested non-biosolid based products and biosolid based fertilizer products. The top half of this table here is non-biosolid based products, and here's all the different PFAS in there, and you can get a rough estimation of how much they found, right? And then the bottom half of the table is biosolid based products. This J product was the one I highlighted just 2 slides ago, and this N product is maybe a little more representative of those Class B biosolids I had mentioned before. And as you can see, there's a wide variety of concentrations, even some of the biosolid based products not having nearly as much as others. And then there's products that have nearly 100 parts per billion PFAS in them, which is a lot more. All right, so once you apply biosolids to the land, what happens to them? Well, over time, they leach out of the actual granules or biosolid product to become available in the soil, right? And once they're in the soil, they're called forever chemicals for a reason, because they effectively don't break down, at least not on any time scale that's relevant to a human's life. And so from there, they're in the soil, and as Wayne had explained earlier, they get taken up into plants and they accumulate there. From there, they can be directly eaten by people, or they're fed to livestock, and then that is then eaten by people. Additionally, there's typically waste product from agricultural systems, which is often gleaned by wildlife or intentionally fed to livestock, like something like corn stover stalks, right? When a field is harvested for corn, they take the seeds, and if it's intentionally given to cattle, they take the stalk too, and they give it both to cattle. But how much of that soil PFAS actually ends up in the plant and or the animal? Well, it really depends on the plant and the PFAS, as Wayne had kind of highlighted before. The more water-soluble PFAS tend to end up more so in the plant material, and a lot of it tends to not actually reach the seed, right? So if you're looking at something like wheat or corn, you're not going to have as much in the actual seeds. That being said, as I stated before, that doesn't mean it's the only thing that animals eat, and we eat animals, right? So in the end, it's a complex system, and there's multiple ways it gets into people. There's multiple ways it gets into animals, and then after it's in animals, it gets back into people again. And yeah, so that was my presentation. And here's my contact information if anybody wants to get a hold of me later.
Thanks, Dr. Lissay. I just wanted to pause to see if, if anyone had a question that relates to this presentation and this really nice overview of, biosolids and how they're not all equal, which is a really important point for us to bring out, especially as we talk about, the right to know for farmers, right? It's not that all biosolids, may be bad, right? So, and there's a lot of nuance here. So just wanted to pause for a moment to see if any questions are coming in. Maybe Dr. Lissay, you could just monitor the chat where I'm trying to do the same as typing in
Yeah,
answers as they're coming in. And
I'm
then maybe,
not seeing any yet, but one thing I thought about maybe that might be a question is, should we be concerned about all food and all biosolids? And I think I highlighted that maybe not all biosolids, but reference dosage, which are exposure values. Basically, if you go above that amount, it could be a problem. For a lot of PFAS, they're very low. And so while I don't think it's generally a concern for every piece of land that's ever had biosolids on them, I think the threshold for what could be of concern is actually quite low. So do I think every field that has had biosolids applied to it is permanently contaminated in a horrible way? Probably not. I do think there are a lot of them that are, though, and I think that we haven't found most of them. And if you are a farmer, I know there's a USDA grant that if you contact the USDA, you can get a grant to actually test your field. So that is something that I thought it was worth mentioning here.
yeah, that's really valuable. I don't know if maybe at some point there's a link that you could share for that. That would be really interesting to learn more about. But one question from Barbara just came in. DEQ records show that the majority of landfills in Virginia dispose of their leachate in sewage treatment plants. Can you characterize differences between sewage plant biosolids, between the plants that take the leachate and those that don't? Do you know anything about this?
Yeah, again, it's another one of those things. It depends on what's in the leachate. Some of the landfills might be fine. I imagine a lot of them aren't, and so I haven't. No studies are popping to mind, but my assumption would be is that if you have an additional source of PFAS versus one without that additional source, your overall amount is going to be higher. And landfills are also a persistent source of PFAS. A lot of them are highly contaminated.
Just to follow up to your point that you made before this question, is there any support available for farmers that might find via the grant mention that their fields are contaminated to a concerning degree? This is an excellent question and not at all out of scope. Please, y'all, keep putting these questions in here because this is such an important question, right? Why would I ever want to look on my farm to find out only the bad news, right? And yeah, maybe you could touch on that a little bit more.
Yeah. So the idea of what I don't know doesn't hurt me. Well, no, it's been hurting you, and it will continue to hurt you, and never will stop if you don't do anything about it. What to do after you find biosolids in your soil? That's kind of the $1 million question, and there are tons of people working, as Wayne had mentioned, on techniques for treating soil. But frankly, the first thing I would do if you find you have significant amounts of PFAS in your soil, your farm, is to get a hold of a lawyer.
And thank you, Stuart, for posting that link to the USDA money for PFAS soil testing. If you're on your phone or whatever it is, maybe we can compile this information for participants later on, just because that's a great resource there. William asked, has the EPA set acceptable levels of PFAS and biosolids to be spread on agricultural land? And we may have river keepers answering that. And so maybe we'll just jump over to Dr. Berger for now. But yes, we will keep, this is a great active chat. This is exactly what we were hoping would happen in this conversation. So please keep all of these great questions coming. All right. So All right. Dr. Berger is a professor of chemical engineering and biomedical engineering at the University of Virginia, and his lab uses biotechnology to solve important health and environmental challenges, including PFAS. So I'm assuming many other things in addition, and seeks opportunities to translate these technologies to market. And Dr. Berger is a co-founder of CSO of Lighthouse Technologies, which provides biosolutions to growers and producers to improve the health, quality, and safety of our land, water, land, and food. So hopefully you may have some suggestions in your presentation to that prior question. Dr. Berger, over to you.
Yeah, thanks. And thanks for the opportunity to speak tonight. Yeah, I'm happy to participate. So today what I wanted to share was some recent work from our lab, kind of at the beginning of a project that we're working on collaboratively, looking at methods for detection and mitigation of PFAS with a specific focus on food, water, and soil. So this is just a little summary of the lab. Thank you for the nice introduction. We work broadly in a lot of different areas, using biological tools to come up with sustainable alternatives to kind of environmental and health challenges. So antimicrobial resistance is actually something that in a way kind of touches on this topic. We work a lot on recovery of critical minerals or other things sustainably, biomanufacturing, alternatives to chemical manufacturing. And then today I'll talk about our work looking at detection and removal of PFAS in agriculture. So this work is a collaborative effort. I want to make sure I highlight my collaborators. This is truly a team effort. This is a huge challenge, as I'm sure you all can appreciate. It really takes a very diverse collaborative team to try and address a challenge of this scale. So we're in year one of an EPA STAR grant for the next 4 years that kind of allows us to do this work. So this is myself and Mike Timko at University of Virginia, colleagues at Maine, Presque Isle, Connecticut Agricultural Research Station, Central Aroostook Water and Conservation District, the Micmac Nation, and many others. So it really, truly is a very citizen scientist, university scientist, government scientists, we're all working together. So I'll touch on these three things. The first is developing tools as sentinels for monitoring PFAS on farm. And then trying to use that to try and implement strategies for management and mitigation. And then also beginning to look at how it spreads, say, in food webs or other culturally important plants. So things that may not necessarily be agriculture focused specifically, but other plant types that also are very important that people and animals come in contact with. So I like to kind of use this as a motivation for the work. Randy is one of our collaborators. He's the director of Central Aroostook Soil and Water Conservation District in Aroostook County. So he's a farmer as well. This is a picture of his farm. He labels his products as, PFAS broccoli, PFAS Brussels sprouts. That's kind of a blow up of the pictures there. Because he and many others irrigate their crops with water from the Aroostook River, which contains, it has been demonstrated contains significant levels of PFAS. And so, it's kind of one motivation for this work. It's to work with farmers and growers like him to assess the spread of these in soil and crops during a typical grow season. So this is from 2023, kind of prior to this grant starting. So we have, now at this point, we're not quite done with data collection for this year, but once we are, we'll have at least 3 years worth of data to start looking at trends. This is one kind of snapshot of that data that I like to share as an example of kind of outcomes or deliverables that we're trying to give to growers. So Northern Maine is famous for its potatoes. The potato growers are, a major constituency up there. And so, this study focused specifically on the red Norland potatoes, which are cultivated in Aroostook County in Northern Maine. So what we were looking at was different water sources that were used for irrigation. There's 3 listed there. You can see levels that are at or exceeding limits for drinking water in the parts per trillion. And then we began to look at the distribution of these in the actual crops themselves after harvest. And so what was interesting to us was that primarily in the edible parts of the plant, the tubers themselves, we didn't see an appreciable accumulation. However, in the leaves or the vegetative parts of the tissue where there's more water present, we did see accumulation of these. And so as an outcome, this was a report that Randy put out, and we're working towards publishing this more comprehensively over multi-year. Recommendation one could give is that in principle, the edible parts of the plant are are may not have PFAS exposure, but you don't want to plow under that vegetative tissue. You want to make sure that you segregate that and keep that from getting put back into the soil where it's going to accumulate and propagate. So yeah, it's just kind of a snapshot, an example of the type of work we're doing. Another part is trying to develop tools to actually detect in the field. And so these are publications that I have listed. They kind of talk about the technology. We've made these, in short, sort of engineered biosensors that fluoresce or give off a signal, and that signal indicates PFAS binding. And so we're in the process of trying to implement these and commercialize these now. The whole idea is that this is not going to replace advanced techniques like LCM SMS, where you're going to get really comprehensive chemical information. Farmers and growers need something simple, fast, at point of contact to give them a leading indicator of whether there's a problem or not. That's the sentinel idea that I mentioned before. And if you can provide that tool, you can then begin to sort of hone in on where the real problems might lie, or also use it as a tool to monitor transport and fate in real time and really begin to look at sort of broader dynamic transport during a grow season. So this work, again, was done at land adjacent to the Loring Air Force Base in Aroostook County. So the star kind of shows you way up in the northeastern part of Maine. This was a industrial, sort of military activities that were at that site that required storage of hazardous materials. So it's important to emphasize there AFFF, aqueous firefighting foam, that's a PFAS source, but there's a lot of other things in that soil too, PCBs, pesticides, solvents, fuel. So it's a much more complicated thing than just, PFAS in isolation. And so these are found in ground, surface water, as well as soil and sediment. And so this is early work. This is a publication from last year, kind of showing a first validation of sort of the early stage of this biosensor in practice. So here we were sampling different surface water sources that were used for irrigation or were known to be contaminated. We compared our results to the results that we got independently from mass spec. And we saw overall pretty good agreement between the two. And what we kind of learned from this was that the sensors are really kind of much more specific for PFOA, and you can kind of see that in the results here, that really the signal is primarily contributed by the detection of PFOA. So we're not really picking up signal from, say, other molecules that might be present, which is going to be the case in this. But in a way, this is kind of like a leading indicator of sort of the generality of whether or not we're going to see PFAS compounds, say, in a water sample. And so since this study, we've now, been refining these, trying to expand, multiplex it to other types of compounds, as well as, beginning to work with others to look at, meat, dairy, processed foods, and soil, and other kind of solid matrices. And so the idea here is that we don't need to do any extensive sample pretreatment. We can kind of drop this in and do a pretty rapid test. And that's sort of where the work is at the moment. So thanks, and happy to answer any questions that folks might have.
Awesome. Thank you so much. It's always wonderful to see that kind of close collaboration with farmers and research and, local organizations, because really that's where we're trying to really find solutions. And so when we pair that research with it, it's inspiring. I'm going from the bottom up because these questions are coming in so fast. So Mike just asked, what's the cost per sample for a biosensor?
Yeah, great question. We're in the process of developing it. It's, I would say, at least an order of magnitude, or 10 times cheaper than, kind of the average cost we pay to go use a third-party service for LC, MS. Yeah, that's where it's important to work with farmers, because you kind of need to learn where they're at. And, for them, they're operating on pretty thin margins. So, they don't really have a lot of cost to spend on this. So, I think if we're really successful, we can probably drive that cost down to, say $1 a sample or so. And that's where a lot of the kind of commercial activities are happening at the moment. So yeah, it's certainly cheaper and faster, but to how far we can push that is kind of where we're at the moment. So.
And just because Mike's on the same side saying, thank you, how long does it take to get the results back? Sounds like you know the customer.
Yeah, a matter of minutes. Like this is a direct detection method. So it's, and the publication that are there. If you want to contact me or email me separately, I could share those papers with you that actually kind of illustrate all of these details. So it's a fluorescence-based detection method. You can use a portable handheld device. You can attach something to your iPhone. There's a lot of different ways it can be done. And so, a matter of minutes. The time is really the prep work for the sample. And that's sort of another aspect of what we're trying to push, especially as we've gotten into food as well.
Do not underestimate the labor for sample prep is
Yeah,
always.
always going to be a challenge. Yes.
All right, just for another one for you, Brian, and from Barbara. At another presentation today, a researcher characterized the occurrence of PFAS in soils. I'm losing my space here. On Maine farms as unique and due to industrial biosolids from the paper mills. Interesting. And this not comparable or relevant to situations where residential sewage treatment plant biosolids are used. Can you comment on potentially the paper mill inputs from Maine being at play here or not? Or maybe that's.
That's a good question. I would say certainly where we were located in northern Maine, for example, the profiles that we saw are kind of the fingerprint we were getting from the LCMS-MS detection. Certain compounds present in there indicated that likely those compounds were associated with pulp and paper processing. So yeah, that was just sort of anecdotal as we were like accumulating and collecting the data. Um, as far as like residential or other sources, most of the work we've done has really just been trying to find that leading indicator like PFOA or PFOS as sort of a general indicator, and then following up with the LCMS-MS. I don't know how far we'll be able to go with what we're doing, but um, it would certainly be interesting if we could potentially develop, say, a fingerprinting tool to help tell us what source it comes from. But that's that's a very ambitious goal.
I will be reading those papers coming
Yeah.
out of your sure.
Yeah, happy to share.
Okay. Um, so one from Dean Knox, our Potomac River keeper, as this issue gains more attention, it seems major investments in PFAS destruction technology needs to occur. Wastewater plants in Northern Virginia are moving toward incineration of biosolids. Can you talk about PFAS biosolid incineration and other potential destruction technologies? Anything promising? And that may be open for you, Dr. Berger, or anyone else.
Maybe
Yeah,
I sneak in
yeah, go
first.
for it. Yeah, no, that's
Um,
definitely one for the audience.
so when I was a child, I had a teacher who he's of Swedish descent, and he constantly bragged about Sweden's wise use of their garbage as they incinerated it for energy. Well, Scandinavia has a huge PFAS problem right now, likely because the garbage they incinerated just sent the PFAS up into the air and it rained down on them. So there's PFAS all over the place. So my concerns with PFAS incineration always lie with, did you actually destroy it? Did it leave the flume? Is it still there? Did you just transform it into another PFAS? Yeah, that's all I had to say.
No, I would agree. I mean, I think as far as it's a big challenge. Like, I think, well, as the other speakers, everybody pointed out, that's the strongest bond in organic chemistry. So it's not trivial to try and break it down. And so partial incineration could maybe create products which may be worse than the less volatile parental compounds themselves. So it's, as far as I can tell, it still seems to be an open question, and there's not a fully mineralized outcome yet that's really like fully scalable. I'd love to be wrong though.
I mean, it truly is. It's you're
Yeah.
changing its form and moving it somewhere else if you're not truly breaking it down or destroying it. So William has a really important question in the chat here because this is not simple stuff. And can we just talk a little bit about how does PFAS and the different types of PFAS, right? We talked about long chain, short chain in Dr. Magabri's presentation. How does PFAS move in the soil profile? Sorry for the sirens behind me.
Yeah. Typically, more water-soluble PFAS move more quickly through the soil. That makes sense because water is what brings things through the soil. And so the larger PFAS, like like PFAS, like PFAS and PFNA, PFDA, tend to stick higher in the soil column and also closer to the surface. Then it gets to the problem of unknown PFAS. The current 1633 EPA method tests for about 40 different PFAS, and there potentially are thousands of different ones. And a lot of these other PFAS break down into those PFAS that we are currently concerned about. Not that we shouldn't be concerned about the other ones, we probably will be in the future. And I try to explain it like this, is that when you have conversion, one chemistry is different from the other. And so I describe it as, if you drive out to the desert and you find a car broken down out there with its wheels removed, do you wonder how it got there? Well, clearly at one point it had wheels. So some of those less water soluble PFAS might end up in places you don't expect, because at one point they were more water soluble and then they broke down. And then the opposite happens too, right? And so less soluble PFAS break down into more soluble PFAS. And as a result, PFAS concentrations, as an example, seem to increase over time in contaminated sites, especially with AFFFs. Well, it's because things are breaking down into PFAS. And I had some friends at the EPA do a study that showed, 13 breakdown routes of different PFAS chemicals into PFAS. And it gets very, very complicated.
Yeah, but really thank you for explaining that, because I think it's really important especially for farmers to think about, okay, so maybe certain PFAS are draining off my fields, but maybe certain ones are sticking around. Will those get up into the plants I'm growing? Really, we're gonna if we can't truly eliminate these, how are we figuring out how to live with them? So I really appreciate that explanation, because it's not, it's they all behave differently depending upon their solubility. I wanted to get to Stuart's question. We have 3 minutes left, guys. You guys have so many great questions, and we will figure out a way to answer them even if we're past time here. But Stuart's question is an important one. The sludge industry is always on the lookout for situations where the PFAS contamination came from somewhere else other than biosolids, to say, right? So if you find it on someone's field, you say, oh, it just came from the rain, right? So biosolids aren't the problem. We can keep doing this. We can keep, putting this on, selling this to farmers. So, is there any way to connect the work that you all are doing and to specifically say this problem on this farm is because sewage sludge was spread here? And this gets to the fingerprinting question, Ryan. And just if we could talk about that for a moment, because there's some really slick salesmen out there trying to sell this stuff.
Yeah, I think Stephen's talk earlier kind of highlighted sort of in some sense, what the profiles look like, right? So that kind of fingerprinting type analysis. So I guess usually when someone says something like that to me, I kind of point out it's kind of a problem no matter where it came from. So, and that it's coming from biosolids because it's likely to have had it. And so you're kind of introducing it as a problem. And so I don't know. Yeah. I don't know, maybe someone else has a better answer than I, but yeah, I think it's kind of on that it's already a problem. So kind of move on from that argument.
So PFAS are completely anthropogenic, which means they're solely made by humans, right? They don't exist in nature. And so, as Brian had mentioned and Wayne earlier, like you can track down sources, right? So PFAS doesn't just magically appear in places. And when it's coming from the rain, that does happen, but concentrations tend to be much lower and they tend not to match up exactly what a source might be. So there is potential there, but yeah, like we had talked about, source tracking, find what was applied there and test it and see what you find in the actual site.
Oh, try to get off mute there. We are at
the cost to have biosolids tested for PFAS, typically between $500 and $1,000 for the analysis. You might need extra help for collecting the samples and then also interpreting them. But yeah, and as I had mentioned earlier, there's grants out there, and you might be able to find nonprofits who will help you.
And we're always
Uh,
looking for folks to work with to do this type of testing. So if there are folks who, especially because this is more of a regional chat, we're always looking for ways to kind of help validate, improve, learn, So that's, that cost was such a big driver of why we pursued what we did, was that just was prohibitive for a lot of people to be able to do. So
yeah, and prohibited for the research too, right?
yeah,
It's
yeah, yeah, exactly.
But this has been such a rich conversation. I apologize that we can't get to all the questions in the chat. But I just want to mention that this is the first of 3 webinar series, and the next one will be on Wednesday, December 17th, focusing more on policy solutions to PFAS and biosolids. We will hopefully take this chat and maybe be able to put together. Your questions are really good because they help us understand what information do we need to get out to you more. And so stay tuned. But yeah, just to say that I apologize that we couldn't get to them all, but thank you to all of our panelists for being here and for this community. I think the community and the power of this comes from people really banding together and getting on the same page, basing it in good science and really going forth with advocacy and solutions from there. So great work, everyone, and hopefully see you at the next one.